Okay, we are recording. Start. Let's see what that's about. Okay, anyway. Let's do some attendance. Bangura, not here. Casebolt is here. Trisden is here. Covington Griggs, not here. Kaylee, not here. Dow, not here. Hughes, not here. Lebdy, not here. Carla's here. Mankins, not here. Mathis is here. Rains is here. Reed is here. Elise, not here. Thompson is here. He went to get tweezers and graph paper. We forgot that. But other than that, it's good.
You just shove this back there somewhere out of the way. That's why it's on there.
Sure.
It's working.
And we have the, yep, got it. All right, yep, thanks.
All right, sorry about you.
It's okay. And finally, Yindui is here. Elkins is here. Well, it's 10:01, so I'm going to close and lock the door. As a reminder to anybody watching this video, got to be present. But as I've kind of been hinting at for the past week, this is the worst semester I've ever had as far as like people getting pre-labs done. And as you can see today, people actually coming to lab. And again, I take responsibility for that. So something's just changed all of a sudden. I don't know what it is. I'm going to try to figure it out. So one more thing, I'm going to kind of relax the rules a little bit. In that, usually I said, if you miss the lab, you get 0 credit, right? So now I'm saying, if you miss the lab, you can still do it for half credit. It's like a handful of people, a lot of people aren't even here. So hopefully you've taken some initiative and you're watching this video at a later date, and you're going to do the lab yourself and get your half credit. And that being said, to remind you, I'm here to help. So when you find yourself doing this lab, you're doing the lab, but you need help doing it, meet me. Actually, you know what? I just started, so. No problem. So that's the way it's going to go from now on. If you missed the lab, yes, you can still get half credit. And as always, I'm here to help. And I'm recording this, so hopefully you'll get the data. And also you want to get in touch with your classmates so they can give you the data if you don't get it from the video. Or I can give it to you. So any questions about how the labs are going to work from now on? All right. Was it Elise, right?
Yeah.
Okay. Sure, I'll mark it down that you're here. You didn't miss any important stuff. Well, I guess you kind of did. But as far as today's lab goes, you didn't miss anything important. All right, you are accounted for. All right, so before we talk about this week's lab, just really quickly, I want to give you a heads up for next week's pre-lab. I don't remember for sure. All right, first let me say this. The next lab we're going to do is really important. This is the photosynthesis respiration lab. So this is the one you have to write a lab report about. So since it is so important, what we're doing is next week we're going to meet and we're just going to do the pre-lab lecture. So I'm going to give you all the information to know how you do lab, and then I'm going to teach you how to write a lab report. That way, when we meet the week after that, we can hit the ground running to do our experiments. So because of that, I don't remember off the top of my head if this pre-lab is due this Friday or if it's due next Friday. If it's due next Friday, but you turn it in this Friday, I'll give you extra credit for it. Because, the sooner you do it, the better. That being said, I want to give you a heads up like I've been doing all semester, so please pay attention. When you do this lab, pre-lab, excuse me, pull it up here. You can see here, there's 2 short videos that you need to watch while answering some questions. Then, as usual, you've got your pre-lab questions that prove that you read the introduction. As usual, the answers need to come from the introduction. You don't look it up on the internet, right? So not only do I want to make sure you understand the concepts, but that shows proof that you read the introduction. And then when you're done with the questions, you need to go on and provide your hypotheses, right? And I've made it big and bold and red, so hopefully it's easy to see it. So there's 4 sets of hypotheses, right? So question 1, right, we're gonna do that experiment, write a hypothesis about it. Then we're gonna do another experiment, right? So write your hypothesis about that. Then we're gonna do another experiment. So you're gonna write your hypothesis about that. And then finally, another experiment, write your hypothesis about that. And also, just so you know, like, hopefully you've noticed this too, you can also do predictions for extra credit. Some of you have been trying to do that, very good. Just so you know, prediction is an if-then statement that describes how you're gonna test your hypothesis. So, in this case, for example, well, I don't wanna talk about it too late. I'll just give, if you don't know what a prediction is, I can help you, or you can click on that and then give you some examples, right? And it's extra credit, so my hat's off to you for those people who are trying. So any questions about what's expected of you for next week's pre-lab? All right. Please don't miss it, but again, I guess from now on, I guess if you do miss it.
I do have one question.
Yes, good, question.
You said you're gonna go over the information of the pre-lab in class this Friday or Monday?
Neither. So next week when we meet for lab, we're not doing an experiment. It's such an important experiment.
Okay.
The next week is gonna be nothing but a pre-lab lecture where I explain how the lab's gonna go. But also I'm gonna teach you how to do a lab report too. Good question. And I'll be recording it too, I hope. All right, so let's jump into this one. This lab takes a while, so instead of wasting your time doing what I usually do, which is talk about the pre-lab questions, I'm gonna jump directly into the experiments. So bear with me for this. There we go. Turn the light on. It's a lot of writing on there, but it's really not that complicated, I hope. So hopefully by now you guys have all done the pre-lab lectures, or excuse me, yeah, pre-lab. Yeah, you know what we're talking about here. Can anybody tell me what diffusion is? Basically speaking, what is diffusion?
Diffusion is when a molecule gets too crowded and moves into a less dense.
Essentially, yeah, right? So if I were to light a scented candle, for example, light it up here, first you guys won't smell anything, right? Then Tristan would smell it. Then it's, it would go back and forth all the way to get all the way to the back, right? That's diffusion. There you have small, high concentration, low concentration. So when it comes to heat, if you increase heat, will that increase the speed of diffusion or decrease the speed of diffusion? Increase, good, right? Because heat is nothing but molecules moving fast, right? So that would make sense that would make that too. What about size? If you increase the size of something, will it diffuse quicker or slower? Slower, good, right? Because it's bigger, it just takes more energy to move it. All right, so those are the 2 concepts that we're going to worry about. The first experiment you're going to do, and I'm going to break you up into teams again. So, you'll do one experiment, another group will do another experiment, another group will do another experiment. As a reminder, you still need to answer all the questions, right? So you might be doing one experiment, but you need to answer all the questions. So if you need to talk to your classmates about how they did their experiments so you have an understanding of it, feel free to do that. Anyway, we're gonna break it down into 3 groups. The first group is gonna get 3 of these Petri dishes, 2 of which are plastic, 1 of them is glass. What they're gonna do is draw a line down the middle of each one, and then they're gonna write MB on one side and PP on the other, because MB stands for a dye called methylene blue, and its molecular weight is 320, right? And then PP is the other dye they're gonna use, and its molecular weight is 158. So obviously this MB is a lot bigger than the PP. You see that? All right. So that being said, forget all this, the different temperatures for now. We have 3 different Petri dishes. What you're gonna do is measure them every 15 minutes, and every 15, or as you're doing that, is the black one. There we go. As you might expect, these things are gonna diffuse, right? So it's gonna start off as a very small dot, and it'll get bigger and bigger and bigger as time goes on. Does that make sense? You're just gonna use a ruler and you're gonna measure how big it is every 15 minutes. You with me so far? Okay, so after this is all done, after 60 minutes of this, which one do you think is gonna get bigger? The big dye or the small dye? Like the one that has a big molecule or a small molecule? What should we expect to get bigger? The methylene blue that weighs 320 or the potassium permanganate that weighs 158? Based on what you guys just told me correctly, what you guys just correctly told me. What do you think is going to get bigger? The smaller one, right? Smaller one. So we should expect that one to get bigger because it's smaller. It should diffuse quicker because it's smaller. Does that make sense? So its blob is going to get bigger. And that's all you're going to do for 60 minutes. Every 15 minutes, you're going to measure the size of it and then put it up on that board. Nice and simple, right? All right. So again, you're going to have 3 different petri dishes, and you're going to do that. And you're going to be comparing which one's spreading quicker, the larger molecule or the smaller molecule. Additionally, since there's 3 different ones, your 2 plastic ones, right? One's going to be in the cold. You're going to put it in the refrigerator. One's going to be at room temperature, right? And then you're going to have one on a hot plate, which, if you don't mind plugging it up for me while I'm talking. We got this hot plate. We're going to make sure it's not too hot or it will boil over. Anyway, again, same thing. I mean, we're still just doing the same thing, right? You're just measuring it and putting your data up there. So which one do you think is going to spread quicker? The cold one, the room temperature, or the warm one? The warm one, right? Because like you guys just correctly told me, it gets, the warmer it is, in theory, the quicker it'll diffuse. So that's what you're looking at, right? So again, group 1's going to do that. They're going to measure it every 15 minutes. So the one in the cold, the one in the room temperature, and the one in the warm for methylene blue, and also the same for the potassium permanganate.
15 minutes or 15 seconds?
15 minutes. Good question. Yeah, it's a long one. I mean, really, if you did, it doesn't diffuse that fast. If they did, there'd be something wrong. So yeah, every 15 minutes. Good question. Which is what I was saying last week. You guys might want to bring something to do because there's a lot of waiting in this lab, which is what I'm trying to get it started for you. So any questions about the first activity? All right, activity 2 is fairly simple as well. You're going to have a beaker. You're going to put some water in it. And I'm going to help you, but what you're going to do is you're going to use this stuff to make a cell. This is dialysis tubing. As far as we're concerned, this stuff is the same as a cell membrane. It lets some things in, but it doesn't let other things in. And again, I'm going to help you do this when the time comes. But you open it up, you're going to fold it, you're going to put a clamp on it right here. Of course, I'm doing half of it for you right now to make things a little bit quicker for you. There we go, clamp it. That way, nothing comes out that side. There we go. And you're going to kind of open it up, and you're going to put stuff in it. You're going to use these little droppers to put stuff in it. So if we're talking about activity 2, which is the one I'm talking about right now, this one here, we have this solution that has starch and salt. Make sense? That's what's in there. You're going to put this stuff into this. Does that make sense? That's it. This into this. Then you're going to clamp it so nothing gets out. Then you're going to put that into your beaker filled with water, and it should look like this. Good so far? Okay. So what we know about diffusion, since this is pure water in the beaker, and in here we have basically a salt starch solution, what should we think? Keep it basic. Don't think too hard about it. What does the salt want to do? Do we think the salt wants to stay in there or come out? Should want to come out, right? Diffuse, because it's high concentration. There's a lot of salt in here. There's 0 salt in the beaker, right? So we should expect it to diffuse out. What about the starch? What do you think? High concentration in here, 0 concentration outside.
Yeah.
It's going to want to get out, right? That's what diffusion tells us. However, this is like a cell membrane. So some stuff can't come in and out. We don't know about this yet. We'll see. So let's say some of this stuff cannot come out and diffuse out. What do you think the water is going to want to do? Go in, right? Because it just wants to make things balanced, and that's what we call osmosis. So if the water goes in, we consider that osmosis. If the salt comes out, that's diffusion. If the starch comes out, that's diffusion. So how do we test? How are we going to know if water went into this cell? And you can look at the table over there to give you a hint, or what's on the board. When you're done with this, after however many minutes, after 30 minutes maybe, however long it is, how are you going to know if water went into that thing or not?
You're gonna wait.
You're going to wait. Perfect. If it gained weight, it had to have been water, right? Well, for the most part. Here's where it gets a little bit trickier. How are we going to know if starch came out of it or not? Because if it gains weight because water came out of it, but, well, I don't want to muddy the waters. Here's how you do it. You put some iodine in the water. So what happens when iodine meets starch, as you should know from last week's lab?
Happens when I absorb it.
Yeah, it'll turn dark, right? So if the water turns dark, that means starch must have come out into the water because it is now mixing with the iodine. Does that make sense? If the inside of the cell turns dark, that means the iodine went inside the cell. Does that make sense? All right. And then a similar thing here. We're going to test for the salt as well. Yep. We had this stuff called silver nitrate. Again, just like you did with the iodine, you're going to put some drops into there. And if it turns cloudy, right? So if the liquid in here turns cloudy, that means salt has come out. Does that make sense? So that's how we can tell. We can tell by weighing it and putting iodine and silver in there to find out if starch came out, if salt came out, or if water went in. And then you're going to write it here. In the beginning, there is water in the beaker, but not in the cell. So if it gains weight, you're going to put in the beaker and in the cell, right? Yeah, I'm not going to waste your time talking about all this, but that's how that's going to work. We can help you fill that out when the time comes. Finally, the third activity, this one's very easy too. You do the same thing. You make yourself a little cell. This time, the other people, instead of using starch solution, will use this sucrose solution. They're going to put it in the water. Obviously, if it gains weight, that means water was absorbed, right? So that's osmosis happening. If it loses weight, obviously sucrose must have diffused out of there. Pretty simple, right? So, but for these people, what you're going to do is you're going to weigh it every 15 minutes and put the weight. Does that make sense? All right. Oh, really quickly, just so I don't almost forgot this. Even though it's on the board, I want to remind you. So table 2, whoever's doing activity 2, not only will you be testing it at the end with the iodine and the silver, but you're also going to take a beginning weight and a final weight to see if it gained or lost weight. Does that make sense? Okay. The math, how to do the math right here for total change is right there. How to do percent change is right here. I think that's it. And really quickly with reminders, when you're done, make sure you upload the PDF to my online. But again, like I've said last week, people are having problems with that. So however you get it to me, we'll figure something out. Again, do the entire lab, not just the experiment that you do. You guys are working in teams, so you're going to share data, but that's it. Don't share anything else. Right? Don't copy off each other's answers. Make your own graph, and ask for help. Now my kid's sick, so once I get you guys started, I have to leave, and you'll be in his hands. But that doesn't mean you can't come get. Again, let me help you. Meet me online. Meet me physically in my office. I can help you do all these things. So, any questions? All right. Once we'll break it down again. Really quickly, do group 1, group 2, group 3. Yep, group 1, group 2, group 3, group 1, group 2, group 3, and put you on group 2 since that's more people for that. They'll need more help. All right, so you guys go to your groups, get started, and we'll be here to help you. Did you have anything to add before they get started? Okay. I'm going to keep my hands on this and make sure. If it gets too hot, it's very bad. All right. So group 1, might as well use this table over here. Lane blue on this side. And good. Lasers to do that. Here's the graph paper. I'm gonna just put it up here, I guess. Graph paper if you need it. I mean, you'll need to do a graph, but it's up to you if you want to use graph paper or use the computer or whatever. So group 1, everything you need is right here. And once I get group 1 started, then I'll start with group 2, and then I'll start with group 3. Not that you guys need to wait on me, but. All right, group 1? All right, so if you want to go ahead and draw it like that, you know, put the line down the middle, put MB on one side, on the left side, and PT on the right side.
This is just flat.
Yeah, just like that, yeah. Right, sure. That's- Oh, yeah, we'll get that drink. Sorry about that. I've opened the door. Like I said, unfortunately, this lab has a lot of waiting. So while you're waiting, if you want to go outside, call somebody, have a smoke, get something to drink, whatever you want. I don't care. Perfect. Good. So what you're going to do now, I'm going to show you how to do it on this one, and then you can do it for the rest. So this is the cold one, right? Once it's done, you're going to put it in the refrigerator or the freezer, even better. So the directions don't call for this, but I'm going to do this anyway. When you get this, it's like a gel. I don't know if you can see it, but it's a gel. So my suggestion is to poke a little hole in it. Yeah, see that little hole? This way, because historically this is the hard one. I see a problem with this. This should help things. Now that you got that little hole, it's going to be really hard to do, but you want to get these tweezers, put a very small amount of the glue on the obviously the methylene blue side. Just make sure it adds up, right? Methylene blue and B. Try to get the smallest amount you can into that little well that I just made, right? If you're going to do that, and then obviously the potassium permanganate, the same thing. And then put again that one in the freezer. This one you can just keep out. Yep, this one the hot plate. And every 15 minutes, you just take your measurements, like it shows in the lab. You literally use the one measure in centimeters how wide this little blob is. That's it. All right, number 2, right? Yep. Help you guys. First of all, I'm gonna make sure that's on.
It's a great way to check.
Yeah. I would say you guys shouldn't do this because, you know, you're valuable. I'm not. I'm just an employee. Anyway, all right. So for you guys, we're already started on yours. Actually, if you're group 3, you might as well come watch what I'm showing group 2 because you're doing the same thing as far as making the cell is concerned. You're just using a different solution. All right. Actually, I'll let you guys do it. It's the whole purpose, right? So, I'm going to hold that, please. Or unless someone else wants to do it, whatever. I need one of you to use this to get it in there. All right. And Tristan, can you just put, I don't know, 100 ml of water, unless it says otherwise. I think 100 ml is good. So it's for you guys. You do the same thing, right? You're gonna group 3. You're going to use these scissors. Where'd it go? Oh, he's got it. There you go. I'll just go ahead and cut it for you.
What is that?
This is the dialysis tubing. So as far as we're concerned, this is a cell membrane. It's gonna let some stuff in, but not other stuff. So whoever wants to do this, here's your 2 clamps, group 3. You're gonna wanna fold it, clamp one side, and we have extra. So if you guys mess one up, we have extra. So you guys just grab it. Sure. It's probably everyone's first time doing this, right? You could expect some mistakes. So like I was showing them, I'll show you, more like this. You have a bit, or folded in there? Yeah, perfect. Okay, folded like that. So he's folded it and we're gonna clamp it. That, and then what we're gonna do, you're gonna do what I showed them. So you're gonna, I've got the stuff you need. You just need to get one of those little squeezers. Right? Yeah, that is the proper term, yeah. You're gonna put as much in there as you think you can, but obviously, when you're done, you can do what I've already done, which is fold it, clamp it. And then, we can just, pure water, and they have the pure water, so you get it from there. So water in here, put this solution in this cell. Now with those, Are you, yeah, you zeroing it out?
What do we weigh it? Grams?
Grams, good question, yes.
Do we need to get some water?
Yeah.
Oh, and that, let's see, you don't, yeah, is it working? Good, all right, can you put that on there, please? You get the initial weight, correct?
We haven't gotten the zero.
That's okay, yeah, I'll show you how. There you go. Now, hit that button, push it until it goes to zero. There you go, now you're zeroed. Wait, I see what you're saying, I'm sorry.
Yeah, we need to weigh it.
Yeah, good grief. No, okay, I'm sorry, you're fine. I gave you the wrong, I was thinking of a different experiment. Just clear it out again. In this case, you don't necessarily need to zero it, right? Because what you've got, when you weigh it, it's gonna include the clamps, everything, right? So nothing's gonna change other than the water that might be coming in or the other stuff that might be going out.
You want us to pour it or just?
I think
water.
the pipe out would be the best way, but, wait, for the water? I'm sorry, yeah, water you can just pour in there.
Yeah.
Yep, back there.
Are these okay, or do we redo it?
Yeah, you're gonna wanna redo that one.
Yeah, sorry.
It's okay,
This one usually? What's
it's
it?
better. It's hard, right? It's hard to get that little bitty amount. That's good.
Now what's next?
Better, I should say. Now you do the same thing, but the other dyes. So that's the methylene blue, and now you can use the potassium permanganate, which is over there too, but you can.
Oh, okay, that's why. Thank you. I'm sorry, I'm listening to you. I want me to walk.
So, no, it's okay.
I just-
So for you guys, if you were writing a lab report about this, which you're not, but if you were, like, one of the things you would talk about is how the blobs are inconsistent, right? So they're not even starting the same size. So your data is gonna be a little bit off because of that.
That should be good enough.
This doesn't have an option for grams, so we're just gonna have to convert it.
Should, it says 600 grams.
Yeah, no.
So what's it giving it to you in milligrams?
Pounds.
Pounds? What? In the world?
I think it should be fine. It's in the picture.
Yeah, that's the button that should be doing it.
It switched from ounces.
It doesn't matter.
It's actually fully under the water.
If it can go wrong, it will.
No, we just need to open the other with the clamp part and see what
The world. I might have to get another, might use the other.
that's.
There we go, percentage. This shouldn't be the hard part of the lab. There we go, it's in grams now. But very good on all from you guys to notice that. It's very easy, something that's easy to
So
overlook.
initial 100.
So guys, when they're done weighing theirs, you should probably use their scale because fix theirs and it was kind of a pain. Might as well benefit from it.
So do you just pour it? It says from activity 2, question 19. So I gave up last time, I was filling out for the 2.
When you say nothing, you say something.
And I'll also give you 5 points of extra credit
Yes, it's supposed to. But
if you email me a picture of the data when everything's done. So I figure if I bribe you all, at least one person will do it for me, because your classmates who aren't here are gonna need that data, and unfortunately, I won't be here.
So when it expands, it's gonna go to the wax or it's gonna be like on top of the wax?
Oh, it's made out. You know what I mean? Like it'll, should get bigger
It's gotta be pretty.
like that.
Like it'll eat up the wax or not? Just keep pumping.
Yeah, you know what I mean? Like that blob will be the same shape, but bigger. I don't know if that makes sense. You'll see.
Like, so it's not maybe just a little part or the whole?
Oh, got you. I would just measure the main blob.
Okay, I missed that whole part when you said measurement.
Okay.
You might need a little more water, I guess, just to make sure it's fully submerged. I mean, the directions probably give you a specific amount, but as long as it's enough to be completely submerged, you're good.
like clip and all, or just the
Yeah,
plastic?
that's a good question. All the way up.
We're doing the same thing that they did.
Exactly,
Weigh it
yeah.
First.
Exactly, except instead of using starch and salt, you guys are using sucrose.
Oh, okay.
And they have to, and instead of like doing all the different color changes and stuff, you guys are just doing measurements, but they only do measurements twice. You do it every 15 minutes. Kind of balances out.
We just want to write.
Oh, so this is the sucrose you put in there?
The secret is what he's putting in his cell.
Is this where?
Oh, that- oh, so we're putting the sucrose in the water.
Or what?
Okay.
Now you just need to clamp it.
Yeah, clamp it. And we gotta weigh it first.
Stop recording now.
I forwarded the
Okay, okay.
Sorry.
That's all right.
It's like,
How do we know if water diffusing in the cell gets to the inside of it and gets dark?
Good question. So the water, if it goes into the cells, that we call that osmosis. And the only way you can tell is if it gets heavier. I mean, technically,
It won't
right?
change color?
No, it's not just from the water alone, right? Because, water and starch won't make it change color. But
It's like 0.7.
iodine, like he's doing right now, that, as we learned from last week's
I'm sorry.
experiment, when iodine and starch mate, they get like a dark color. So if it becomes dark, we know it's starch. Technically, if it gains weight, some of you could argue that maybe it gained weight from the iodine or it gained weight from the silver. But it's such a small amount that probably that's not the case. Did I keep this overnight?
All right, you can.
There you go. And I think you only do this towards the end, if I remember correctly. But yeah.
So
All right.
Set a timer.
And I'll show you what I mean.
So do we take,
Now,
we
yeah,
exclude the
now it's a
clamp
weight
weight?
game. But so here's the starch solution, right?
How do we exclude the clamp weight?
Oh, it's just accounted for, right? So
Yeah.
that's 180.
118.9.
Yeah, right. That's how much it weighs now, right?
30 minutes.
Clamps and all.
Oh, okay.
So whatever it gains or loses, it's pure.
Yeah.
Yeah,
18.9.
but I'm glad you're asking.
18.9 grams.
All right, so here's what happens, just as a point of comparison. When you add the iodine to the starch, see,
Yeah.
right? That's what it looks like when it mixes. So if your beaker turns that color, you know starch must have come out of your cell and got into the beaker. Meanwhile, if this stuff gets into your cell and the cell turns that color,
We don't have to
you know
drop
that the
anything.
iodine got into the cell. Does that make sense?
Ours is just weighing it.
And that's your point of comparison.
Yeah.
So we just gotta do a lot of weighting.
And up a little.
Yeah, please. You're gonna wanna hold your breath and make it quick. It's a stinky freezer. Yep, freezer. Yep.
Hold your breath.
Yeah, it stinks bad.
Oh.
I don't know what's in there or what used to be in there. There's nothing in there now.
It's that Dahmer.
What?
The Dahmer.
Now, if the iodine diffuses in the cell, then it'll change color.
Yeah, the cell will turn this color if the iodine gets in there. But if the starch comes out, then the beaker will turn that color.
Right.
And of course, if neither one of those happens, then that means the starch didn't move and neither did the iodine.
It stays
Yeah,
clear.
yeah, exactly. Yeah, or that this color. Best way to describe it. All right, I'll show you one more thing too. Just again, as a point of comparison. This is sort of like your positive control, right? That's your positive control for iodine. Now you know what to look for. I'm gonna show you what to look for with salt. I want this in the instructions, but these are already so complicated, I don't want to make things worse. Because that's pretty cloudy to begin with. Here's what it looks like when you put silver iodine in. Oh, see? It's milky. Yeah. So again, when you do put this in there, if the water in the beaker looks like that, then you know salt came out. And of course, if the cell itself looks like this, you know that the silver went into the cell. That make sense? Yeah. All right. But just so you guys know, because again, you'll have to answer the questions on it. This is what it looks like when silver mixes with salt. It's kind of hard to see unless you come over here, which you don't need to do right now. Feel free to. It's cloudy. And this is what it looks like when iodine meets the starch. So this is how they're going to be able to tell. If the starch comes out into the beaker, it'll no longer have that yellow color. It'll look like this, right? But if the iodine goes into the cell, then the cell will no longer be cloudy. It'll look like this, right? And of course, if the salt comes out of the cell, it'll look like this. Well, it's kind of hard to see now, but so that's basically how that works. And of course, if it gains weight, we can assume water went into it. If it loses weight, we can assume, the salt or the starch came out.
The activity on the unshelled eggs, or is
Oh.
leaving that blank?
Yes, yeah, you can completely leave the shell thing blank. I listened to your feedback and you guys unanimously, everyone who voted said skip it, so we skipped it. Which I think is a great idea. It's a messy, nasty mess. Excuse me, I'll be right back.
I spent a time.
Yeah, there's only two things that I do.
So it's an hour altogether. But
Yeah, everybody has an hour.
Did you bring your paper that he has to sign?
I did. Yeah, at first I was like, hey,
Hey, Sean said we're cut if we don't. Oh, well. Must be nice.
Creative filters,
You play sports?
the activity, is that, are we just gotta do this?
We just gotta get the
We don't have to do,
we have to answer the questions, but we didn't do it.
what about this one? I thought we're not, this.
You only do doing this, for the
Yeah.
lab, as far as the paperwork is concerned, you're doing everybody.
Okay.
Hey, when's our first day after spring break?
Friday.
Oh, this Friday.
Oh, wait. Nick,
Is our next game tomorrow?
Yeah, got no work.
Your first or?
oh, okay, yeah, Friday.
That's your first game?
I thought tomorrow was Friday, but yeah, Friday.
Is it a conference or just?
Yeah. Is that conference?
We don't
think so.
Who?
Bluefield?
They're not at NBC.
Yeah, they're not.
They're in West Virginia.